Professor Paul Long, we as a faculty of Life Sciences and Medicine, particularly enjoy our inaugural lectures. It's our moment and opportunity to celebrate the academic achievements of members of the faculty. And as such, we see this as also a great opportunity for those faculty members to acknowledge the support and the advice and mentorship that they have had typically during their academic journey. Just a few words about the faculty before I turn to Paul. The faculty is one of the 8 faculties of the university. It's actually the largest of the faculties, which means that, and indeed a very successful faculty, and that translates into us having a very active inaugural lectureship program. We, as I mentioned, enjoy that by virtue of an opportunity of celebration, but it does, of course, also indicate that we are and have become a wonderful environment for our academics to really develop their careers. Faculty is broad. It embraces medicine, biomedical sciences, and pharmaceutical sciences, of which, of course, we will hear more this evening. So, may I turn to just a brief introduction and a reflection which I'm sure will be expanded on by Paul in his lecture. But Paul's academic career started, of course, as is true for so many, with his initial undergraduate degree here in London. He then undertook an MSc in microbiology at Birkbeck, also in London, before moving to Cambridge for his PhD. He then went to the States for his initial postdoctoral training and fellowship, before returning to London and his first academic appointment at somewhere called the School of Pharmacy, which I gather is also here in London. That's a joke, by the way. Should there be anybody from the School of Pharmacy here. And indeed made his first advance in promotion from lecturer to senior lecturer whilst at the school. He then probably made the most crucial and important decision, that was to move across here to King's, and was appointed as a senior lecturer within the Institute of Pharmaceutical Sciences. Then advanced and was elected to a readership in 2012. And, of course, that trajectory indicated it was inevitable, and indeed has now come to fruition, that he would achieve election to a full professorship, which took place in 2016. So it's a great pleasure to introduce and invite Professor Paul Long to give his inaugural lecture, which, of course, rather ironically, is entitled Sun, Sea, Sand and Me.
Well, good evening, everyone. Thank you all for coming, and thank you, Professor Trembar, for your kind introduction. I don't think there are many situations in your life when so many people from so many different eras of your life turn up at the same place at the same time. But this is one of those situations, so thank you all for coming. It's really fantastic that this has happened, albeit a little bit daunting, I have to say. People ask me often, what is it I actually do? And I struggle to answer that sometimes, because over the last 30 years or so that I've been involved in scientific research, I've picked different techniques from lots of different disciplines. So it's hard really to pin me down, but if I was to sort of like pick the writing that runs through my stick of rock, I'd have to say that I'm interested in finding new medicines from nature. So this is natural product discovery, also known as pharmacognosy. And I think ever since man has crushed grapes, we've been interested in looking at nature as a source of new medicines. And, that goes back to antiquity ever since, there's been historical writings. But more recently, natural products being used as medicines has been recognized as well. For example, here in the 2015 Nobel Prize, which was jointly awarded to Yu Tu in China. And basically what she did was to go through all of the historical records of ancient Chinese medicines to look for any plant that had been used as a possible treatment for malaria. And the hematologists in the audience will recognize the typical ring forms here of the malarial parasite. So from her work then, the Chinese team isolated artemisinin, which is an anti-malarial drug. So she shared the Nobel Prize with Campbell and Omura for their discovery of avermectin. So avermectin then is used as an antiparasitic for the treatment of roundworm infections. For example, you can see here then river blindness, but also elephantitis as well. So this is avermectin. And they isolated it from a soil microorganism called streptomyces. And streptomyces are bacteria that I'm going to come back to later on during my talk. I would like to say that Campbell's and Omura, and also Yu Tu, are also pharmacists by training. And Campbell actually is a graduate at the University of Wisconsin, which I'll talk about also a bit later on. But the discovery then of antibiotics and other compounds that we use in medicines from soil microorganisms has its history. This works. Here we go. A bit too far away. Has its history really with the discovery of penicillin by Alexander Fleming back in the early 1930s. And so you can see here then the penicillium fungus producing penicillin then, which is destroying these staphylococcal bacteria. And this then led then to a golden era in modern drug discovery, using microorganisms as a source of compounds. And this was really led by this guy here, Selman Waxman, who coined the phrase antibiotic. And he discovered some 15 antibiotics, including streptomycin, which is still used today as a frontline drug in the treatment of tuberculosis. So both Fleming and Waxman also received Nobel Prizes for their work in natural product discovery. So there's a long history then of Nobel Prizes being awarded in this area. I'm going to return to some of these drugs, for example, erythromycin and penicillin. Penicillin is one of these compounds that seems to be haunting me all of my career. So then I started thinking about, well, an inaugural lecture. I didn't start out to become a professor of marine biotechnology and therapeutics. It was kind of like a journey that seems to have happened. So I tried to think about, well, where did I get my inspirations from? And I think my very early inspirations have to come from my mother and from my father. And my mother, who's sitting up there in the audience, she comes from a long line of wet fish merchants in Grimsby. So my first job then was actually on Grimsby Fish Market, working with my grandfather. And we basically had to go buy the fish, fillet the fish, and then send them off to the distributors, which usually are Greek fish and chip shops in London somewhere. And the first thing he said to me was, you're going to get very cold on the fish market, so you might as well put your hands in a bucket of ice. And that was enough for me. There was no way I was going to follow this as my career. On my father's side, he comes from a very large family of Irish, and they like their alcohol. He was a landlord. Okay, so I actually grew up above several pubs in South London owned by the Young's Brewery Company. And you can see the Castle pub here in Tooting, South London. So I grew up in the Castle pub in Tooting. And Tooting is a location, again, which I don't seem to be able to get away from, which I'll talk about a little bit later as well. So I grew up then in Tooting, South London. I went to school in Tooting as well. Family circumstances meant that I could only study for 2 A-levels, and I have to say I did them very badly as well. So I was limited then where I could go and do my first degree. I didn't qualify for university just with 2 A-levels. Okay, so I had to go to Polytechnic. So I went to the Polytechnic of Central London. And I read for my first degree in biotechnology, which in those days, this was the only institution that offered that degree in the whole country. And I really cut my teeth, I guess, you know, first taste of science at PCL, working on penicillin. Okay, so they basically specialize in the biotechnology department on the genetics and fermentation of the penicillium fungus. It was a great institution actually to be at because sort of like in the 1980s, people who can remember what was going on in higher education in the UK, Margaret Thatcher was the Prime Minister, Sir Keith Joseph was the Minister of Education. So it was a time of turmoil in higher education. And certainly at the Central London Polytechnic, the student body was very fierce. Lots of protests and things like that, which I actually quite enjoyed as a young man. So yeah, the student's voice is something that, you know, I take very seriously, and it's still something I'm very passionate about when I'm at King's as well. So that was my first taste of science then, was penicillin at Central London Polytechnic. From there, I had a brief séjour in the health service, and I worked at St Mary's in Paddington. And guess where I ended up? In Fleming's old department in clinical bacteriology. So it's here on the second floor where Fleming made his first discovery on penicillin. And this really was an eye-opener for me, because this was the first time when I really came across real people with real infections. At that time, in the late 1980s, St Mary's was a big treatment centre for HIV/AIDS, and of course at that time there was no treatment at all for that infection, so people were dying. So we were seeing people presenting with very unusual infections and then dying as well. So, you know, real people with real infections and a real need for new drug treatments as well. So this really stimulated my interest in drug discovery as well. I'm often asked to review grant applications for the research councils, and when it's got to do with antibiotics and new antibiotics, you always see in the impact statement words like, you know, there's a desperate need for new antibiotics. And I often wonder, you know, do the authors really understand what that means? You know, how many people really appreciate that, you know, real people do die of infections? I'll be very honest with you as well, working for the health service is not the greatest paid profession either. So I left as quickly as I could because I couldn't afford to stay. At one point I was even thinking about moving into the nurses' home because I had to do a lot of locum work in order to pay my bills. And so I moved to Glaxo Group Research, based in Greenford in West London. And this character here, Howard Wildman, was my boss, and he was really inspirational to me. His name is Wildman, and he really was a wild man, wild by name, wild by nature. He's Australian, you know, so he was a very garish kind of character. And he was really inspirational to me because he actually taught me how to do science. You know, he taught me what an experiment was, how to actually set up an experiment, how to design it, and what the results actually meant. What does statistical significance really mean? And most importantly, Howard taught me how to publish in the scientific literature. He taught me how to publish papers. So I'm really grateful to Howard, and he's looking forward to seeing the YouTube of this lecture as well. I told him all about it. But at Glaxo then, I joined the natural product discovery team, and my job was to isolate these very unusual Streptomyces bacteria and fungi, and then to screen them against a whole variety of different disease targets. And this was a really interesting time in drug discovery because it was at a time of the computer revolution. The World Wide Web was invented. All of a sudden, we had personal computers, not laptops at that point, but we certainly had internet and we had computers. No mobile phones then still. When I talk to the undergraduates and I say, you know, there was a time when there were no mobile phones, they look at me like really strange, you know. But yeah, we still had no mobile phones here at all. But it was a computer revolution, and so of course then came robotics as well. So this allowed us then to move from just screening maybe a few hundred organisms a week to several thousand as well. There was also a revolution actually in the pharmaceutical industry itself. It was at a time then when pharmaceutical companies started to merge with each other, or there were aggressive takeovers, depending on how you look at it. And Glaxo was no exception, okay, at that time. Glaxo merged with Burroughs Wellcome. And I took that opportunity then, that merger, to take voluntary redundancy. And the package was really good. In those days, it was excellent. There was no cap on redundancies at that point. So I used that money to part fund my PhD. I should say that while I was at Glaxo, I did an MSc in microbiology part-time, 2 evenings a week for 2 years at Birkbeck. Quite why I decided to do that, I have no idea now. I mean, it must have been madness to think that you could work full-time during the week and then go to night school. It seems crazy now. I don't know how I did it or why I did it. But anyway, Birkbeck was a good experience. And also what it means now, though, is that I've experienced all levels, really, of tertiary education in the UK, from polytechnic to night school to the University of Cambridge. And that experience, I think, is really good when it comes to actually dealing with undergraduate students. Here at King's, for example, we have mixed ability teaching. We have students from all different backgrounds, students from all over the world. So myself having the experience of going through all the different types of institutions, I think I have a certain empathy for the kind of undergraduates that we get and their needs and desires and hopes and things like that. Anyway, at Cambridge then, what a place, you know, what a place. I mean, the dreaming spires, you know, the Gothic architecture, the opulence of it all, you know, it was a very opulent kind of place. And I matriculated here at Clare College in Cambridge on the backs, and I did spend quite a lot of time on the backs as well, doing a bit of punting, I have to say. The master at the time was Bob Heppel, Sir Bob Heppel, the late Sir Bob Heppel, who's the well-known civil rights lawyer who was Nelson Mandela's lawyer in the 1960s. And he gave me a useful piece of advice. He said to me, get to know your PhD supervisor, because your PhD supervisor could be your friend for life. And that certainly has been my experience as well. This is my PhD supervisor, Professor Peter Ledley, and he's been really inspirational to me throughout my career, but also in my personal life as well, you know. He's just a great guy, just to be able to pick up the phone and just shoot over any kind of problem. And he always has some kind of advice to give me. So it's been a great pleasure, you know, knowing Peter, you know, professionally and also as a friend as well. What did I do with Peter? Well, Peter was working on lots of different types of antibiotics that belong to this group called the polyketides. And he was particularly interested with this antibiotic, erythromycin A, produced by these unusual Streptomyces organisms, these soil organisms. And what Peter found was that erythromycin A was actually produced by a very, very large protein, actually a series of proteins that form a complex. And what he found was that each of these proteins could be divided into individual catalytic domains. And each one of those domains catalyzes part of the reaction leading to erythromycin A, or the A-glycone precursor at least. And you can see here that the molecule is color-coded, and this corresponds to the different parts of the protein that catalyze the reaction. So essentially, it's a production line, like you'd have a car production line. It's exactly the same kind of idea. So what Peter thought we could do would basically be to reprogram the DNA that codes for the protein. And if we could reprogram certain parts of this enzyme production line, then we could alter this molecule in a predictable way. In other words, we could design new antibiotics. So my project really was to look at the beginning of the biosynthesis, the starter unit. And part of the project was funded by Pfizer in Groton in the United States. And one of Pfizer's big drugs, of course, are these macrolides, such as azithromycin or Zithromax. And they produce this antibiotic typically for inner ear infections for children. Okay, so penicillin, again, is the antibiotic of choice, but of course, if an individual is sensitive to the penicillin, then they have to have an alternative. And the alternative is the next generation erythromycin, such as Zithromax. So they were interested in what we were doing, and they part-funded the work. And it was successful. We managed to actually produce a compound in high yield for them that went into clinical development with Pfizer. The spoon, I'll come back to in a few minutes. The spoon is important, okay? The spoon is important, okay? So this was very successful, and my PhD work was published in Nature magazine, which for any scientist, they'll tell you that that is really, kind of like the highlight of your career if you can publish in Nature magazine. Everything after that then goes downhill. So I started at the top, and then what was going to happen? Well, failure happened, actually. Failure happened next. I'll talk about the spoon in a few minutes. I'll come back to that. But what happened next was that I moved to the University of Wisconsin, to the School of Pharmacy, and I worked with the late Dick Hutchinson here. Again, he was an eminent medicinal chemist. And I spent 2 years then, and this is the research group. Everyone here now is either in academia, professorships themselves, or they're in very high-ranking positions in the pharmaceutical industry. I notice I'm the only person who's got a drink in my hand there, but I guess it's Christmas. Anyway, what happened there in Wisconsin was that nothing worked for me. I was actually sponsored by Julian Davis, and for those of you who know Julian Davis, he basically really is the godfather of modern antibiotic research. Back in the late 1950s, early 1960s, he was also at the University of Wisconsin, where he discovered that antibiotic resistance genes can be transmitted from bacteria, one bacterium to another, and then also to human patients as well. So this gave us then a mechanism of how antibiotic resistance spreads in the community. And so this was also at the University of Wisconsin. But Julian had a spin-out company called Terragen out of the University of British Columbia, and he funded my postdoctoral research. Julian is still alive and still active in the lab. Okay? But he gave me some really important advice a few years later when we bumped into each other. He said, well, how's it all going? And I said, well, I think it's going fine. And he said, well, what about grants and things like that? And I said, well, I think I'm running at about a 10% hit rate. And I said, I thought that was a bit low. He said, well, can you imagine if 10% of your experiments actually worked? And through Julian, I learnt the concept of failure, because my 2 years in Wisconsin, nothing worked for me. So I went from Cambridge, publishing in Nature magazine, to Wisconsin in 2 years of absolute failure. So I learnt that science doesn't always work. Okay? And actually, in my lab today, we have this saying, we dare to fail. Okay? We dare to fail. What I also learned from my time in the United States was the concept of competition in science. Okay? I always thought you go into science and everyone plays with a straight bat and, and everything is all fair and equal. It's not. There's a lot of competition in science, and the Marquis of Queensberry rules don't always apply. And this is something that I learned. And I learned from Julian and from Dick Hutchinson how to do science properly and ethically as well. Okay. Back to London then. I moved back to London, and I can't get away from penicillin. And I'm very happy to say that Mike Sharland is in the audience. So Mike Sharland from St George's in Tooting. Again, I can't get away from Tooting, can I? And when I arrived back in London, I started to teach undergraduates in pharmacy, and I was teaching them all about antibiotics. But they kept on asking me the same question. Yeah, it's really interesting, these antibiotics, how they work and stuff like that. But how do we actually use them? If we have a patient in front of us, what do we do? So I suddenly realized there was a big gap in my clinical knowledge. And Ian Wong from UCL School of Pharmacy invited me to join this burgeoning group called ICAP, or Improving Childhood Antibiotic Prescribing, which Mike started, and we're still going strong. We're a bit of a loose group of people. Some people come, some people go, that kind of thing. But our overall aim is to start looking at prudent prescribing of antibiotics in children. And Sonia Saxena, she's a GP and also academic at Imperial. She's a key member of our group too. Now, the medics in the audience will recognize what you're looking at here. So this is a tympanic membrane, or an eardrum. This is a normal eardrum, and then this is where we start getting infection. Okay? So infection of the inner ear, or otitis media, or acute otitis media with effusion. So this is when the eardrum then bursts and then leaks out the fluid. It's the most common reason why a child will go and see a GP in the UK, and the most common reason why a child will receive an antibiotic. And that antibiotic, believe it or not, is penicillin. And what we wanted to know was, where does the dosing guidelines for penicillin in children actually come from? What do I mean by that? Well, what we found was that, looking at the BNF, which is the British National Formulary, which is the guidance used for prescribing in the UK, basically children were put into different types of age bands. And these age bands then corresponded to changes in dose. What actually happens is that you can get a big child will receive half of the adult dose. A small child will receive half of what a big child will get. And then a baby will receive half that of a small child. So the dosing regime then in the UK has been just to halve the dose from an adult all the way through to a baby. And we thought, well, where did that come from? And looking in the literature, this is where the spoon comes in, okay? So a child, particularly a young child, is not going to tolerate taking a tablet. They have to have a liquid formulation. So you use a spoon to do the dispensing. So the spoon typically then will be 5 mils. If you turn it over, it's 2.5 mils. It's half that dose. So this is where this halving comes in. And what we found was that these age bands were first introduced back in 1963. And in children, they haven't changed at all. They've changed in adults, but not in children, despite an increase in weight of children because of obesity and antimicrobial resistance. So the relative dose children are getting is actually very, very low. But does that matter? So we decided to have a look at the dose and, what is the clinical significance, if at all. So amoxicillin then is the type of penicillin that's frequently used or prescribed for children with acute otitis media. And in the UK, we do actually have some guidance here. It's recommended that children receive 40 mg per kg per day. United States is much higher. It's 90 mg per kg per day. And if we actually look then at the age in years of the children and work out their dose, mg per kg per day, you can actually see that the majority of children are not receiving anywhere near what the recommended dose actually is. Okay? And if you're 5 years and above, it's about half the recommended dose. So we're putting children into age bands and they're getting really, really small amounts of the drug. So in effect then, are we actually treating the infection? We published this work in the British Journal of General Practice. So this is the journal then for British general practitioners. Essentially, we were criticising the GPs, and I was expecting a huge backlash from these GPs, but they actually gave us an award for best research in paediatrics. Shortly following on from that, it's nice, the National Institute for Clinical Excellence, who published with the Department of Health this BNF, the British National Formulary, then changed their guidance. So our work, and Mike and Charlotte is in the audience as well, our work has been directly translational and of value, I think. Our research has led to a change in UK antibiotic guidance for children. And that can affect something like 5 million prescriptions each year. So I think that's quite significant work that we did. Okay, so here's an aerial view then, showing all the different sites that King's College is on, just for anyone who doesn't know it. And when I came back from the United States, I was still interested in my natural product work. I was still interested in looking at these unusual soil microorganisms as a source of antibiotics. And this is where my ex-supervisor, Peter Ledley, came into the picture. He said, don't do it. Competition is too fierce for funding within the UK, because UK is a centre of international excellence for these type of natural products. He said, do something else with your scientific career. Of course, I wasn't very happy when he said that, because I had my heart set on doing this. So I had to think about it. And I decided then to move into the marine biology area and start thinking about microorganisms in marine environments and microorganisms that are associated with marine animals, marine invertebrates, and also marine plants, seaweeds. But we're in London, so where am I going to do marine biology? Down the River Thames? I had to pick a different research site for my field work, and that was Australia, the Great Barrier Reef. Oh, I'll go back. Okay, the Great Barrier Reef. And so I was based in Townsville at the Australian Institute for Marine Science. And I was lucky enough to spend, I was working it out, probably about 3.5 years of my life in Australia over a period of about 10 years under various different fellowships and awards. And this is an absolutely fantastic research facility. Not only do you have world-class laboratories and accommodation as well, but they have direct access to the Great Barrier Reef because they also have research vessels. So we can actually go onto the boat and then out to the reef for extended periods of time. We can go for 7 to 10 days, typically. And here's just some shots of what life on board the ship actually is like. And you can see there are laboratories there, not much health and safety going on. There's drink and food and everything, and no lab coat either. Remember, we're in the tropics here. This is Chris Battersill. He's now at the University of Auckland. It's a working boat, so the back of the boat, we're not just doing experiments, but we're hanging up dive gear and things like that. This is Walt Dunlap, and he's featured really heavily in a lot of my research. And here I am here doing some kind of experiment. I'm not too sure what I'm doing. And then you can see me here just in the living quarters. And some of you might recognise this gentleman. This is Marcel Jaspers, who's a medicinal chemist from the University of Aberdeen. And again, we've done lots of work together. So what work were we actually doing? Well, the clear waters of the tropics are clear for a reason. There's not much nutrients in that water. So sessile invertebrates such as coral and sponges and the like, they have to survive somehow. And they survive by taking up a symbiotic relationship with microorganisms. So they live with microorganisms, plants and bacteria and fungi. And that's how they survive. And in surviving, they actually generate this ecosystem which higher animals also survive on, such as fish, Nemo, for example. And this diversity is huge. It's actually estimated that there are more species on the Great Barrier Reef than there are in a tropical rainforest, say in Brazil, for example. So this is a great environment then to look for natural products for these chemicals that we might use then as brand new medicines. So what kind of work were we doing then? Well, lots of these natural products from marine invertebrates, from sponges, for example, have gone into the clinic and they're used. Nearly all of our drugs that are antiviral have their origins in marine natural products, every single one of them. Okay? Acyclovir, all of the HIV drugs, they all come from marine invertebrates. Now, because so many of these invertebrates are associated with microorganisms, I asked the question, is it really the animal that makes these compounds, or is it the microorganism? If we look at some of the compounds, and this compound here is called patelamide, it looks very similar to some of the compounds that we see from terrestrial bacteria. So my hypothesis was that actually it's the microorganisms and not the animal that makes the compound. And to test this hypothesis, we were looking at the patelamides. The patelamides are in preclinical studies at the moment. These basically reverse tumour resistance for vincristin and vinblastin, okay, themselves natural products. And it's produced by this organism, this Aclinum patella, which is an ascidian, also known as the Indo-Pacific sea squirt. And it's green, or it's greenish-blue colour, because it contains this organism, a microorganism called prochloron. And the reason why I chose this particular example is because prochloron lives within the cloacal cavity of the animal, so essentially in the stomach of the animal. So to get the prochloron out, all you do is squeeze the squirt, and out it comes in relatively pure amounts, okay? No contamination with the animal. So we had pure DNA, and I actually did this on the boat. So I cloned the DNA, the biosynthetic pathway leading to the patelamides, and we managed to actually isolate the patelamides then when we expressed this DNA in a heterologous host. So this was the first proof then that the symbionts of marine invertebrates are the true biogenic source of natural products that we can use as medicines. The fact that we could express this DNA in a host that we can grow in the laboratory also got us around this problem of supply. Everyone was interested in the patelamides, but it means you had to go to the Barrier Reef or the Indo-Pacific region and harvest this animal, which is, in terms of conservation, is very unfriendly. But we now had a way of doing it in the laboratory. So we had two firsts here. And we weren't the only people. A group in Utah and a group in Oregon were also doing the same work, but Brotherhood of Science and all that, we published together. Okay, we did publish together. Here's another example. This is an ongoing example here at King's. This compound here called chondroxin A. This was discovered by the National Cancer Institute, active against osteosarcomas, so bone cancer. And what we basically did with Walt Dunlap and Agri Grigiardis from our dental institute was that we collected the sponge that makes chondropsin, which is called Acinia ramosa, and we got enough material to take this compound then into preclinical development here at King's. And what we actually found was, actually, we weren't interested in osteosarcoma so much. We wanted to know what effect it actually had on a molecular target. And what we actually found with Aggie was that we had differential activity on bone remodeling. So the cells that are responsible for laying bone down and then taking bone up, okay, there was differential activity. So this now is in preclinical development here at King's for the treatment of osteoporosis. And this is ongoing work. Now, when you're working on the reef, if you're away for 7 to 10 days, there's a lot of downtime. You know, if you're diving all day, you have to have a certain amount of time to get that nitrogen out of your tissues, and you can't work in the lab all the time. So there's a lot of time when you can just stand at the back of the boat and just look out at the open ocean. And when I usually go to the Barrier Reef, it's during the whale mating season, or the time actually the whales give birth. And it's a magical time on the reef for this, because we moor the boat actually in a lagoon, and this is a sandy area, and the whales love this to give birth in. So for several days you'll have a pregnant whale just swimming around, making lots of noise, as you might imagine, and then all of a sudden the whale will disappear. And within a few hours, then up will pop baby and the mother. And these calves are really, really interested in the boat for some reason. They will swim around the boat for several days. And it's really magical because you can stand at the back of the boat and you can just look eye to eye with a baby whale. So it's quite a magical experience actually to do. And I was doing this one day, you know, I was just, you know, waiting for a while. They call it whale watching, I call it whale waiting. It take a while. It was hot. Remember, we're in the tropics here. And I felt myself getting very, very hot. And I felt my skin burning as well. And at that time, it was very low tide in the lagoon, and the corals were also out of the water. And the corals are animals like we are. So I just asked myself the question, well, if I'm burning, are the corals burning? And why don't they get sunburnt if they are burning? And the answer to that is that they make their own sunscreening compounds. And this was known, these compounds known as mycosporine-like amino acids had been known for a while, and they had been implicated as being sunscreens, but no one had ever proven that there were photoprotective activity in these compounds. So Walt Dunlap again, and also our colleague Malcolm Schick from the University of Maine, we undertook some research to look at the biosynthetic source of these compounds, but also, did they have any photoprotective activity really? So here's a blow-up picture really of a coral, and coral itself is made up of many thousands of individual animals called polyps. And here's an example here just of a nubbins of coral, and there are many hundreds of individual animals that you can see here. And corals belong to the same group of animals as the jellyfish. And you can see here that, not surprisingly then, they have tentacle-like appendages, just the same as a jellyfish would. But they're sessile, they're in the Great Barrier Reef, the clear waters of the tropics, and so they have to be associated with a microorganism. And this microorganism is known as symbiodinium, and it's photosynthetic. And you can see the symbiodinium in the tissues here. So the coral survives because of the photosynthesis with the prochloron. What we found in our studies, actually what happens is that the prochloron produces a compound that it passes over to the coral animal, and the coral animal then converts it into a sunscreening compound, into these MAA compounds. And we called this shared metabolic adaptation when we found this out. It was known that these MAA compounds can actually pass up the food chain as well, into fish, for example, and also into other invertebrate animals as well, such as squid. But at that time, it was thought that this was just applied really into the tropical regions. What we then decided to do was look at a much wider range of different types of fish and invertebrates. This was an undergraduate student project, I have to say. We sent our undergraduate students to Billingsgate Fish Market early in the morning to look at whatever the catch was. And they took a variety of different fish from tropical and temperate waters as well. And in every single fish that we looked at, in the light-sensitive tissues, so in the skin, in the roe, and also in the eyes as well. This actually comes from a dolphin eye. We actually found a group of compounds, these MAA compounds, but always a group of the same 5 compounds, always the same. So there must be some evolutionary reason for this. But the compound that's always in the highest amount is this compound here called palathine, or palathine, however you want to pronounce it. And so we decided to look at this compound then for our experiments to see, well, do these compounds, are they really sunscreens at all? Are they sunscreens? And this is just a schematic just to show you the kind of damage that sun can do to the skin. And I thank Carl Lawrence, who's a PhD student I share with Anthony Young, who's in the audience right now. So here we have skin cells. There we have the epidermis and the dermal layer here as well. And here we have the sunlight. And of course, sunlight can be very beneficial to us in the synthesis of vitamin D. It can also have very bad effects as well. It can directly damage DNA or any molecule that's going to absorb the UV radiation, and that can have several effects itself. It can damage the DNA, so certain essential genes may be turned off. You may build up mutations leading to skin cancer. But also as well, you can actually turn genes on. You can start producing proteins that will have effects that the ladies in my life always ask me about. How do you get wrinkles? Well, what happens here, of course, is that we actually turn genes on that break down the connective tissue in the skin, leading to the wrinkles. You can have photosensitizing effects as well. So basically, the UV radiation will produce these reactive oxygen species also as well, or free radicals, you might know them as. And again, these have several different functions. But what we do have, though, in the laboratory, and this is in Anthony Young's laboratory, and Anthony is in the audience too, we have tests for all these different types of damaging effects of UV light or solar light. And so what we can do is then we can expose human skin cells to solar simulated radiation, and we can add our compounds, these palathine, these MAA compounds, and see the effect. And what we actually found was, surprisingly, that this palathine compound had protection, or gave protection, in every single area that could possibly have photo damage. And this is really unusual, because if you go to Boots and pick out a sunscreen, there are several different active ingredients. The reason why there's several different active ingredients is because they protect against different damaging effects. But we had a compound now, single compound, that protected against all of these effects. And we patented this, and here's Anthony and here's Carl. Carl provided this at very short notice because he said he didn't like his photo being shown. But I wanted to show it because Carl is a PhD student, and now he has a patent. So he's co-inventor now of a patent here at King's. And this IP has been sub-licensed now to this company called Ethic, and we have these 2 products that are now going into production. So the production line is now being put together in Milan, Italy. And we have the trademark for this, these products called Demagi. And you can imagine we have a day and a night cream as well. So it's an anti-aging moisturizing cream with built-in UV protection. And why do we have a night cream? Because the sun isn't out at night, but the damaging effects of the UV radiation still linger on after your exposure. That's why you have after-sun creams. And the palathine also works to alleviate the effects then of what we call these dark reactions as well. So these are the products that we have hopefully on the market very, very soon. And I just want to highlight Carl really for all his work in this area. Okay, so back to the coral again. We were interested in looking at how these MAA, these sun-screening compounds, were actually produced. And I decided to use a technique called proteomics to look at these biosynthetic pathways. And proteomics is a technique that allows you to look at all proteins in a particular tissue sample all in one go. And with just this gentleman here, Malcolm Ward, he heads up the proteomic facilities here at King's. We looked at the proteins in this particular coral, Stylophora pistillata. I have to say as well, Malcolm and I knew each other in a previous life. He also was at Glaxo at the same time as I was. Yeah? So, the world is a very small place. But what surprised me, though, was when I looked at all of the proteins that came out of our proteomic technique, was that there was a huge number of toxins. And these toxins were related to the toxins that you see in the venoms of higher animals, terrestrial animals, such as, for example, tarantula spiders, centipedes, and certain types of snakes as well. And this had never been shown before, simply because this technique had never been applied before in the marine environment. No one had ever decided to use this technique. So this high-throughput proteomics then uncovered a whole range of toxins in this particular coral that were related then to the type of toxins that we see in venomous animals. And I thought that was really interesting and worthy of further investigation. To do all this kind of work, though, doing all this proteomics, we have to kind of like identify the proteins. So we have to match the proteins that we get from the instrumentation with proteins that we see in databases. So to do that type of matching, this is known as bioinformatics. And I've been heavily involved in bioinformatics for many years with the University of Zagreb. And here's just a picture then of Daslav Ranelli from Zagreb. And I've worked with Daslav now for longer than I care to remember. I mean, a huge amount of time, nearly 2 decades. And that's his wife here as well. This is Walt Dunlap's wife, Rumi Dunlap. Walt came to Zagreb to celebrate his retirement and his 65th birthday. And we'd all been working together for such a long time, we decided to have a bit of a get-together as well. And you know, some of you might be able to recognize what we're eating. We do eat the animals we work with, all right? So that is octopus. The person that is missing from this is this gentleman here. This is John Cullen from the University of Kaiserslautern in Germany. So John, myself, and Daslav have worked together for a very, very long time in bioinformatics. And we also set up our own company called Semgen, really to develop bioinformatic programs, which we make free to the scientific community. So this is a not-for-profit organization. The idea is that we receive donations and research funding to stimulate young scientists in the former Yugoslavia. And that's our motivation for doing that. It's a non-for-profit organization. Okay. Back now to venoms and corals. And I said before that corals are related to jellyfish. And I thought to myself, well, no one had made this connection then between all these toxins in the coral and the type of toxins that we see in the venoms of higher animals. We actually use the toxins in the venoms, say, for example, of spiders, as medicines. Okay? There are several examples where we use them in medicines for pain control, for example. We use it to control bleeding, for example, as well. So I decided to look at the literature, and what we actually found out was that we know quite a lot about treating jellyfish stings. And here's an example. This is the box jellyfish then from northern Australia. And this is potentially life-threatening. People actually die from being stung by this jellyfish. So it's treated as a life-threatening condition. And that scarring doesn't heal. Okay? That's permanent when you get that. So we know a lot about treating jellyfish stings, but actually virtually nothing about their venoms or the toxins that are in their venoms. And I thought this is ideal grounds for natural product discovery. Could we use then the toxins in a jellyfish sting as a source of new medicines? So in the last part of my talk, very quickly, what I'd like to do is to talk about what I'm doing now and a little bit about what's going to happen in the future. And the story now changes to Brazil. Okay? So we moved from Australia now to Brazil and the University of São Paulo in Brazil. Now, University of São Paulo, USB, is a strategic partner of King's College London, so ideal partner for us to work with. And there are 3 people that I work with there. Adalberto Pessoa, who's the Vice Dean of Pharmaceutical Sciences, Gabriel Pagila. Now, Gabriel and I also knew each other in a previous life. We knew each other from Townsville. He was an academic at James Cook University in Townsville, so we knew each other for many, many years before he went back to Brazil. So we were working together on a King's College-funded project, again, working on these soil microorganisms, working on soil antibiotics. And I just happened to mention these proteomic results that we were getting. And he said, well, if you want to work on venoms, next door to us is the world-famous Butantan Institute. And this is the largest producer of snake antivenom in the world. So we said, OK, well, let's go and talk to these guys. And I said, well, we're really interested in jellyfish. And what was their response? Well, we work on snakes. We don't know anything about jellyfish venom, which is exactly what we knew from the literature. But what they said was that there's another guy, Antonio Marquez at the university, who works on jellyfish. You might want to go and talk to him. And so we did. And Antonio Marquez, or Chim, as he's known, is also director of SEBIMAR, which is the Marine Institute for the University of São Paulo. And it's based just outside of a very small town known as San Sebastián, which is a seaside town opposite this island called Ilhabela. And here's the island here. And you can see there's this channel running between the mainland then and the island. And this channel really is fed by fast currents from the Atlantic Ocean. So animals then are forced through this channel. So it's an ideal place to actually to go collecting. And this is Ilhabela, so you can see the kind of location that I'm going to in Brazil as well. This is a view from the laboratory, so it's straight out onto the water, and there I am there. We're just about to go on a collection. This is another PhD student of mine, Paloma Nitel. And basically, to do the collections of jellyfish, we just hire local fishermen to do this on a prawn trawler. And we just take the little boat out to the main boat, and off we go. So this is my commute in Brazil. It's a bit different from my commute from Brighton to London on Southern Rail right now, I have to say. And so we go out trawling. And I get undergraduates from King's to do this. So we offer these kind of projects then to our undergraduates, and they go out and they do a bit of trawling. And here's one of our undergraduates. And basically we pull up the nets, and then we get the undergraduates to sort through the nets, the catch, and pull out all the jellyfish. And this is Lucas Brammel. This is one of our undergraduates. And here's an example now of one of the animals we were working with. So this is Alindia sambaquinensis. So this is unique then to southern Brazil and northern Argentina. With a name like Samba, you expect it to come from Brazil, don't you? It's a really beautiful animal. And again, with Malcolm Ward, we did this proteomic technique, and again, what we found was a huge number of toxins that were related to the toxins in venoms of higher animals, such as different types of rattlesnake, vipers, tarantulas, things like that. So again, this really sort of like stimulated me with the idea that we could use jellyfish or jellyfish toxins for natural product discovery as sources of new medicines. And we decided to go further, not just looking at the animal itself, but the microorganisms associated with the animal. And this had never been done before. So we're at the stage now where we've collected lots of different animals. I particularly like this one. We've isolated the microbes that are associated with it, and we're starting to screen and getting some results. Just very quickly, the type of screens that we're looking at, we can do experiments in Brazil we can't do in this country for lots of different reasons. And some of the tests that we're looking for, here's some examples. So here we have Aedes aegypti, which is a mosquito that spreads a variety of different diseases. One that's very endemic throughout Brazil is dengue fever. And dengue is endemic everywhere, even in the metropolitan areas. University of São Paulo, in that district, the area that I live in, Butantan area, you know, lots of people get dengue fever. The people that I showed you, Gabriel Pagila and Antonio Marquez, they both had dengue. This year, the son of Adalberto Pessoa, he also contracted dengue as well. So it's everywhere. This mosquito has also been found recently to have another virus that it can transmit, and this is the disease chikungunya as well. And more recently, which you would have heard maybe in the news, is that this particular mosquito can also spread a third viral infection, Zika virus. So one bite, 3 possible infections you can get from this. We're also interested in Chagas disease, so this is South American trypanosomiasis, and leishmaniasis as well from the sandfly. Too graphic to show you what leishmaniasis actually looks like. Cutaneous leishmaniasis is a really horrific disease. We're not just interested in infectious diseases as well, we're interested in other illnesses too. And a big project that we have, and the haematologists in the audience will recognize what this is acute lymphoblastic leukemia, a type of blood cancer. So we're interested in the type of enzymes, not the small molecules that the jellyfish or the microorganisms actually produce, but actually enzymes that they might have. And the classic treatment for acute lymphoblastic leukemia is the enzyme asparaginase. So we're looking for new sources of asparaginase from jellyfish to treat acute lymphoblastic leukemia. We have a non-medical screen as well, non-medical. Most cars in Brazil don't run on petrol or diesel, they run on alcohol. And the alcohol comes from sugarcane. So they have a huge sugarcane industry, obviously to produce sugar as well. Cachaça, the alcoholic drink of Brazil, but also alcohol that's used then to run most of the cars as well. What happens though is that when you take the sugar cane and you extract the juice and everything, you're left with the raw material, the actual plant material. This bagasse actually is a pollutant. So there's a lot of work going on trying to find microorganisms and their enzymes that will break down these complex carbohydrates in the bagasse. So we're interested also in doing this. And this green fuel is not just a priority area in places like Brazil, it's also in this country as well. The BBSRC have priority areas in sustainability. Okay. Venoms also have toxic effects on nerves and the neuromuscular system, and this is well known, and you can see a variety of different examples shown here. And we also screen for these. When I was first awarded my professorship, I thought, well, yeah, that's very nice, but what do I do now? You know, what do I do with my remaining time, the time that I have left? And I thought, well, this is an opportunity for me to do something completely different, something that I've never done before. And I've never done any neurology. I've never done anything to do with nerves or nerve cells or anything like that. And King's is in the ideal position because we are the centre of excellence in neurosciences, one of the centres of excellence in the whole UK. And we have lots of people here who specialise in pain as well. So this is something now that I've decided I want to have a go at in whatever time I have available to me. I want to have a look at the effects then of jellyfish venoms on nerves, and can we use these then as a potential new way to treat pain, long-term pain. Hopefully this kind of work then will accelerate me into 2017. And I'm not going to say very much about sources of funding that I get, only that anyone who's new to academia or is thinking about taking up an academic research position and is worried about where they're going to get sources of money from, all I'm going to say to them, or say to you, is take it wherever you can get it. You know, all money is good money. The only acknowledgement for funding I am going to give you is to Delta Airlines. Of course, a lot of my work involves me travelling all over the world, and depending on who pays, you either sit at the front or you sit at the back. And I'm lucky enough to have corporate sponsorship from Delta Airlines, and so when I get on the planes, I always turn to the left, not to the right. So I'm really grateful for that. I just want to finish up by just saying a few words about how I feel about my science. And I think having a career in science and in research is a really unique thing to be able to do. And I often consider it to being a bit like walking across sand that no one else has walked across. You're putting your footprints in the sand where no one else has gone before. And I think that's a great way to spend your working life. Buzz Aldrin put it a bit further by saying those footprints belong to each and every one of us, to all mankind. And it kind of like gives me a sense of purpose to think that some of the work that I do, which is new work, which we publish in scientific literature, at some point in the future might be read by someone on the other side of the world, and they might find it useful as well. So doing research, I get a great sense of purpose from. What I have learnt in my journey over the last 30 years or so is that research is a marathon, it's not a sprint. So again, anyone who's thinking about taking up a career in scientific research or is just starting, I just want to leave you with this bit of advice if I can. Keep calm and take one step at a time. And again, thank you all for coming this evening. It means a great deal to me, and I thank you for your attention. Thank you.
By tradition, we don't invite questions at inaugural lectures. We will be also by tradition inviting you to join us for a brief celebration and a glass of something to follow. But before we close, may I invite Professor Peter Hylands, who's the head of the Institute of Pharmaceutical Sciences, to give a vote of thanks.
Thank you, Richard. I had a management meeting this morning, this afternoon actually, with a junior member of staff who was recently appointed. And I asked her what her long-term aspirations and ambitions were. And she said she wanted an academic career, which is good, but she's in an academic position. She wanted an office like Professor Hyder, who is an ex-head of school, but still works for us. And I think she was attracted by his little drinks cabinet in the corner. But she said she wanted a life like Professor Long. And I think we can see why. I mean, it sounds, you've painted a very graphic and very clear picture of what your life has been and how interesting and stimulating it's been, and obviously not only to our students, but also to the staff. So we are pleased to have this role model. If you do go onto the website of King's College and search Paul Long, there's a technical description which he's given some aspects of his research interests. And like me, he's been, throughout his life, been very fascinated by secondary metabolism and the array of chemical diversity that is produced in the natural world and the uses to which they can be put. And as he's explained, by using experimental and bioinformatic approaches, he's focusing on biosynthetic pathways which have evolved to develop this rich diversity. By studying the biosynthesis at this molecular level, we've learned what Paul has been able to do. But what's not really apparent, though he's hinted at it a little bit, is from this rather bland description, that by focusing on the means that the organisms use to produce these structures, it's opened up to him a funding opportunity from the research councils who are interested in the fundamental science of this enzymology, which is not available to those who, like me, have been very much interested in the end products and the use of them. So he's given that a great deal of thought, and we see, I think now it comes from Ledley, who's advised him to do that, and I think we've seen the fruits of that this evening. And so this is exceptional. I want to emphasize that for a natural product scientist. What is also apparent, because he's been talking about his work on antibiotic dosing, it's a really very clear example of the multidisciplinary nature of Paul's work by working with clinicians, which has, as he's indicated, led to a change in guidelines of the BNF for children, which is a very, very important step, as was Paul shown. Over the last 50 years or so, most infants were treated ineffectively by the antibiotics. And it is exceptional that a laboratory scientist can claim to have changed clinical practice in this way. And so Paul is very much to be congratulated by that. And of course, we offer, he's been talking about our undergraduate offering. That is a professional program accredited by the General Pharmaceutical Council. And such examples of multidisciplinary and interdisciplinary practice is very much applauded and recognized by our regulatory body, the General Pharmaceutical Council, as a fantastic example of good practice. So Paul's work, as we've seen, is a perfect example of working across traditional boundaries. And we're very fortunate to have him. I'm very much aware of the fact that it's only me between you and a drink, so I will detain you no longer. But my final task is to thank Paul for a most enlightening and amusing lecture and congratulate him on his achievements. So please join me in the traditional way of congratulating Paul.